Okay, so for this video on looking at mutations from sequence reads, we can think about different types of information that we get from sequence reads. And we're going to look at that over a few examples more broadly at microbes, not just looking at SARS-CoV-2 data. Well, what we can imagine is that we might have say, a linear reference genome looking like this, and then we could draw different segments that are present, like a segment B here, and then we might have a region A, and then it might be duplicated to have two regions A or even three. So imagine these are all identical, and then a region C here at the end. And if we map reads to these regions, we might get particular types of information that's present from these different reads based on their overlaps between the different locations. So if A is amplified in our particular sample, that means we'll have reads that span from A into A here. So those particular reads will span A into A, the start and the end of A. However, there's certain reads here, like this one highlighted, and this one as well, that span B into A. So they can be uniquely mapped here. Over in this end, we have reads that span A into C and A into C here. So they give us unique types of information to resolve that particular region. So that means if we're trying to resolve this region or we map it onto a reference genome that has a single A, we might be able to resolve the B region properly, but then the A and the A into C region properly. But we might find that the reads in the middle that we have very high coverage. And then what we'll find is that we get a slight drop at this segment here because we have reads that are mapping from A to A here, which don't map properly. So we'll get a kind of our average coverage here will look normal, then a kind of a drop, and then a big pile up like that, and then a drop again like that. And that would indicate that this particular gene region is that there's something going on here that needs to be evaluated more carefully. And that can mean that there's an insertion in the sample. And you'll note that we've sometimes referred to these as indels because if we have a deletion or insertion, it depends on directionality. It's a deletion in the sample compared to the reference means that there's an insertion in the reference compared to the sample, vice versa. If there's an insertion in the sample, that means there's a deletion in the genome. In addition, when we're thinking about prokaryotic genome sequences, we should bear in mind that they may be circular. So if we imagine that region above, and we have our region B here, and then A here, then A here, then A here, then C, and then some other region D here. We'll have the same pattern of reads mapping, except that we just should note that the reads will map from B into D and then to D into C. So sometimes we have to just consider other models other than in linear chromosome and also plasmids recombine. So we might get very different structures that emerge when they recombine. Then we have one region that looks like one plasmid, another region that looks like another plasmid, another region that looks like another plasmid, and so on. When we look more broadly, we can identify particular types of changes that occur for different regions. And that's illustrated here by this diagram that we have a chromosome reference, and that reference might be constructed as A, B, C. But if we have a sample and we have reads mapping here, one might be able to identify in deletions where we have lots of reads mapping to A and lots of reads mapping to C, and then we just find that we have a drop in coverage and then for B, like that. Likewise, we might find we have an insertion E, but that's typically we'd have to de novo assemble to resolve E. So after mapping all our reads to our reference, we wouldn't find any big difference here. What we might find is that there's a change in coverage here, that a coverage change from across from A is normal, then B, then C, and it kind of drops there like that at the boundary of B before continuing on. And what we do is de novo assembly. So we take all the unmapped reads and de novo assemble them alone. And we re-emerge with certain contexts like E. And then we do the same with the map reads and try to see is that if there's a new region here. And we can check that by mapping back our reads to this region And if we find that our reads map right over this region, that would indicate that the region is valid. Equally, we can also have inversions, and obviously we get reads mapping here well at C and at B and so on, whether it's to a sample or to a reference. But what we would find is that there's very few reads mapping from A into B, and then we get a change in the orientation of reads at C and B. So rather than in that the reads mapping from A into C will be absent, and the ones joining this section will have a different orientation. The reads point each other. There's a forward and reverse, that they be changed in effect. And we'd also find that reads that we expect to map from the start of B map to the next region on here, whereas we'd expect them to map into A, and vice versa. Reads at the end of C map into A, like that. So those joins between A and C would indicate that there's actually something present here. So we map those reads, we'd find that there's actually a big change here. We've good coverage, then a drop, then it picks up like that, but then there's a drop again. So that change in coverage indicates there's something amiss. Okay, if we take an example of an indel, or in this case, a deletion in our sample, we can think about having our sample here represented as a series of reads without that particular mutation. So we can draw our sample here, and if we imagine these reads are taken from our sample and they lack a particular region in our reference. So our sample here is completely composed of two regions, A and B, and in our reference genome, there's this missing region that is absent in our sample. So this is our reference genome. If we take our sequence reads, our sample will actually look like this, A and B here, like that, which means that we'll have reads that span A and B like that, and they'll have a particular insert size length. Whereas when we map the reads to the reference, that particular read would be located here, and then a long, long way to its partner over here. And if we repeat that, if we have many reads that span A into B, what we'll find is there's a bunch of reads with very long insert sizes that span this region like that. So there'll be some reads that are inside A that are small, have small insert sizes from A and B like that, where the distance will be normal, but the read spanning this junction will be very, very large. That would suggest there's an insertion in the reference compared to the sample. Equally, if we had our sample like this, and here we are, our sample is D, E, and F, and we have extra reads here. These are paired end reads, badly drawn. We have an extra region E here, and then we compare these to our reference, and reference is simply D and F. What we'll find is that there are reads that span D here well, like that, but the read pair for all these samples at D will be absent. Likewise, for F, the read pair for these different ones will be absent, whereas there'll be normal reads inside F here, like that. And then if we took this sample and did a de novo assembly, we might be able to recover that region E. If you think about an insertion is simply the same diagram as I showed you there. And then we think about more complex variations, such as an inversion. So in an inversion, we may have a particular sample where the reads from which it is sampled face the wrong way. So B here has a 5' to 3' for the reference. That's the wrong way around in the sample. So B is like that. So this is our samples reads. So we've got reads from A that go into the start of B like that, and from the end of B to start of C. And then when we map our reads to our reference, we have to bear in mind that we have A here, then we have B in the correct 5' to 3' orientation. So if we track that first read there, we've got A facing forwards here, pointing, and this is facing toward 5' to 3' direction of B, and it's kind of midway down. So that read is going to face like that, and its pair will actually be over here, and they're both oriented the wrong way. This one is at the 5' end of B, so it's actually pointing towards the 5' end. So you can see here that its partner in C is oriented the same way. So this misorientation will identify the breakpoints that the other reads to span inside B will be normal, spanning inside, C will be normal, spanning inside, A will be normal, but we'll see that the orientation of reads that should normally be pointing towards each other will change for A, B, and C, and that's an indication of an inversion. So that means simply that the 3' end is down here in our reference, and 3' end is 5' end is down here in our reference example. Other variations include read mapping at indels. So in this example, you can see here we have our summary of this issue where we have an insertion in our sample. And in this issue down here, we have a deletion in our sample. And we'll see those because the insert sizes of the reads will be very, very different. Those are more complex structural variations that can occur, such as a tandem duplication of A and A. So we'll see here that the coverage is half the size compared to B. And we'll find some reads that are here in A that go all the way into the start of B like that, whereas most of them should be short like this in C. A dispersed duplication, again, we'll get the same thing, that we'll get normal coverage, a slight drop here, normal coverage, well, half coverage, and then an increase at B, then a drop at A again. And if we have a dispersed duplication and we're mapping to that reference, we'll find reads that map from B into A, as they all span here all the way to the start of A, and reads also that span from A into C. So when they map to the reference, they'll span all the way over there like that. And complement number variant is a bit like a tandem duplication there, except the coverage will be lower here at A and increased here for B and C. And again, that effect for A will mean that we have some reads that span all the way to B in our reference. These are more diagrams indicating the evidence of a deletion. The first one of which shows the classic example is simply that if map reads where to a reference where a region is deleted in our sample, you'll find simply that our reads are aligned there with each other, and then there's no coverage then if I use horizontal lines as our reads mapping at this region. However, if we use split read mapping, we can sometimes override this by mapping across the boundary. So since it's going to do de novo assembly without the deletion to see where the other end of the read maps best. So this slide kind of indicates a summary graphic indicating the different types of changes that are present, where the first item is indicating no change. So the reads map directly to our reference here. If there's a deletion in our sample, we'll find that the insert sizes are longer. If we have deletion, we'll see something like that. Okay, so when we have a deletion, we saw that we said the insert size in our sample is changed when we map the reference, that is much longer. When we have an insertion in our sample, we find that the insert size becomes much smaller in addition to the breakpoint observations. And it's the same for an MGE as well. For tandem duplication, we're going to see that the coverage in our reference is going to be much, much higher for that region before it falls. And we're going to find the read mapping here is going to be strange because when I've reads that map across here like that, between the end and the start, that have or their read orientation is changed. So note that we have changes in the insert size. That's it, size, to the coverage. 3 is the read orientation. If we look at this example, if we've a merging of these different reads, some reads will map from here inside to this segment here like that. So that means the read will map from here all the way around here to that one there. And vice versa, well, reads mapping from here over to this side, so that map from here all the way down to here, indicating that something dramatic has changed. And vice versa for the insertions, that we have a read mapping from here to here, and that will be its orientation will change dramatically. It'll be oriented down here, and vice versa. The other side, a read mapping from here to here will map from here all the way back like that. So for inversions, then we'll see the read orientation change dramatically. So then we can look at things like split read mapping, or de novo assembly as a way of resolving those regions. So if we look at an example where we have a reference genome, a sample genome, and there's an inversion here going from black all the way across there like that, we can see the expected insert sizes will change down below between our sample and our reference. Where the reference they're quite they're rearranged considerably. We might find that the read orientation has changed, but the insert size has not. So we can distinguish those from insert sign changes, which are linked to simple insertion deletions, bearing in mind that it depends on the size of the deletion, that if you have very large deletions and our reads are inside there, we may not be able to detect them very easily, in which case we would have to de novo assemble them or do local reassembly. As mentioned, there's other types of insertions like MGEs being inserted and tandem duplications as mentioned. And this summary slide gives you a good idea of the variety of different changes that can happen. If we look more broadly at sequencing technologies associated with this, we have things like PacBio sequencing, which is a single molecule real-time sequencing, where you basically sequence in a pore-like structure like this, where this is a cell on a plate. And we have something similar to sequence biosynthesis chemistry to allow fluorescence to be emitted, indicating based on the wavelength what base it is. PacBio gives long reads, as does Nanopore. So the Nanopore read profile in theory can look like this, where you get very long reads of over 10 kb and many bases. Bearing in mind that obviously the QC will be important there because some might be lower quality. In terms of their application of Nanopore reads to use, this paper from Lemon et al. 2017 indicates that minions are effective against antimicrobial resistance. And for resolving two different plasmids here, we find that having more reads generates a lot more coverage and gives us a lot more resolution of what's actually present. In the same way, we can think more broadly about integrating not just long reads and short reads, but also our methods. So for instance, if we think we have an expansion here of a particular element that's only present as a single copy in the reference, we can take those unaligned reads after mapping and assemble themselves, or indeed we can assemble the whole sample itself as well, and can compare the results for both. In tandem with this, if we're analyzing a bacterial sample, what we should think of is about the steps that are used. So multi-locus sequence typing means that we take 5, 6 or 7 candidate genes that are well studied, and we track alleles on these genes to see what allele or sequence type our particular sample belongs to within a species. So first we will figure out the species, and then usually we can assign it to a sequence type. So this is just based on 6 or 7 housekeeping genes. So it is just simply a classification tool. Then next we just do QC like we did before moving on to options either to map the reads or to de novo assembly, or better still, do both. Then we, after getting mapping the reads, we can identify SNPs after careful QC. We can also take the unaligned reads and de novo assemble them alone, for instance, to get a plasmid sequence. We'll also take the de novo assembly and gradually get a reference by scaffolding the context and taking the unaligned reads to extend the context and produce a better assembly. And then we can compare that assembly against other reference genomes that are relevant. And therefore, we can link these two results together, bearing in mind that de novo assembly is better for looking at structural variants, whereas that read mapping is better for identifying SNPs. And consequently, our approach is really using the integration of both techniques, one focused on mapping, one focused on assembly. Going back to MLST, what that means in practice for illustration purposes is that we get an idea of what is present. And that's represented by these three samples here. MLST will identify that they're an eye, probably belonging to a human. But genome-wide sequence data will give us information on what much more detail what type of human they are. It will miss some things, obviously, but it'll get most of the things. And then we have other variations on MLST that look at 2,000 or 3,000 genes, known as often called CG MLST, core genome MLST. And that will be more comprehensive. Of course, it will miss things like all technologies. Beyond this, we can think more broadly about how we represent genome sequences. And we tend to focus on this linear model as represented here by the human genome reference, where we can assign SNPs where you have an A inserted instead of a T or an indel inserted here. But we can also think about how having de Bruijn graphs, like the node labeled graph here, where we have a graph laid out like this, where we, for instance, we can think about encoding the A and C here as two different nucleotide options at that particular position. So in addition to de Bruijn graphs, there are many other types of graphs. There are directed acyclic graphs, sequence graphs, string graphs, and others. But what's nice about these graphs is that if we think about the nodes, we can identify conserved regions like this GA here and put them adjacent to other SNPs that are node varying within the population, like this AT SNP here. So it can move beyond simply a linear representation of a genome. And what we can think of is what's called a pangenome. A pangenome is all the genes or DNA regions in a sample collection. So all the samples genes put together. So obviously you should theoretically get to some point where if you keep adding more samples, you don't get many more genes, but that is not always limited for bacteria. Depends on the species. So we can think about our pangenome, and therefore we can think more broadly about how do we represent our genome as a linear structure, or as a more accurately is going to be a messy ball like this of connected k-mers. There's a nice tool you can use down here at de Bruijn graph viewer to view de Bruijn graphs constructed at this website. So you put in these sequences, you'll get a de Bruijn graph like this, where you starting off with ACTAG here, but also ATCTC. So you can see that here, ATCT here like that. And there's other examples here as well that you can, so you can try the tool yourself and get a better visualization of whatever you're looking at. So that's the main message from today. We can identify mutations using read mapping. We can visualize the coverage with IGV. We can infer mutations from reads using a series of information, the coverage, the orientation of the reads, the insert sizes, and then more effective methods, as mentioned in 10.4 on integrated mapping and assemblies. We can do split read assemblies and de novo assemblies as well. So that concludes that part on mutations and reads.